Hi everyone. We're going to go over the background information needed to complete your lactase enzyme activity. And once I go over that information, I'm going to actually walk through the lab with you. You can play and pause as needed so that you can write down the data in your student answer sheet and complete the analysis section. So let's start by mentioning we probably have all heard of somebody who is lactose intolerant. Maybe you are lactose intolerant or a family member is. What is lactose? Lactose is the sugar that is found in dairy products, milk, cheese, yogurts, ice cream, all dairy products. It is a disaccharide, and it's a disaccharide that is made up of two monosaccharides, and those two monosaccharides are glucose and galactose. Now, when we ingest lactose, this disaccharide, our bodies have to break that down into the two monosaccharides if we're going to use that sugar for energy. The way that we do that is by adding an enzyme to it. Our bodies produce an enzyme. That enzyme is called lactase. Enzymes typically end in ASE. That's how we know it's an enzyme. Now, our cells, when they produce lactase, they will bind with lactose, right? So lactase is our enzyme, lactose is our substrate. And during that process, during that chemical reaction, the lactose is going to be broken down into glucose and galactose, those two monosaccharides. Now, individuals who are lactose intolerant, what that means is that their cells do not produce lactase enzyme. So that disaccharide sugar, lactose, stays as a disaccharide sugar as it moves through the digestive system. And because it's staying together as a big sugar, it causes all of those digestive discomforts that people who are lactose intolerant will experience. So we're going to do a lab today to see whether or not lactase actually does break down lactose. We're just going to demonstrate a simple enzyme catalyzed reaction. A couple of key points that we want to remember about our lab is that one, enzymes are specific, right? So each enzyme catalyzes its own specific reaction. And two, enzymes remain unchanged after a reaction. So you can have one enzyme that's capable of catalyzing multiple substrates at one after another, right? Because they remain unchanged. So our two big questions for the lab today that we're going to investigate are, number one, does lactase break down lactose? How are we going to know this? We're going to test for the presence of glucose, and I'm going to show you how we do that in a second. And number two, does lactase break down sucrose? Sucrose is another disaccharide. It's what you guys know as table sugar. How are we going to know? We're also going to test for the presence of glucose there. So we're asking two questions here. All right. So I'm just going to move my camera here so you can see my lab setup. And I tried to set up my samples right in front of the beakers or containers that I have drawn down at the bottom. So right here I have my control. That's just a container of water. This is milk, right? So that's going to be my source of lactose. This is also milk, but I'm going to add lactase enzyme to it. And over here in this container is going to be sucrose. Now all I did was take some table sugar and I'm just going to add some water to it to make a sucrose solution. Now, after we after we add the enzyme to these things, we're going to be testing for the presence of glucose, and we're going to be using these little glucose testing strips, and they come like this, and we read them as, we're not going to pay attention to the numerical values. We're simply going to look for a color change, and you can see that originally, the way that the strips come out of the bag is they are yellow, right? 0 glucose. So what I did in this cup here is I actually made a glucose solution. So I took some glucose and I dissolved it in some water. So I just want to show you what it looks like when we get a positive test for glucose. So I would dip my strip in here and I'm going to just wait a little bit for a color change. And it's already starting to happen gradually. I'm going to let that sit there and change a little bit more. It's changing a little slowly. Let me talk to you about how I made the enzyme. So for our source of lactase enzyme today, we're actually going to use lactate tablets. And you can see it says right on the packaging, lactase enzyme supplement. So this is just basically a tablet of enzyme that you would take with your food if you were lactose intolerant, and you'd basically be, supplementing your body with the enzyme that it doesn't make in order for you to digest those dairy products. So all I did was take one tablet out of there and I crushed it down into this powder, and I'm going to add some water to that to make a solution that I can add. I'm just stirring it up now. I'm going to make a solution, an enzyme solution that I can add to my containers. Let's check on our glucose test strip, and let me show you the color change because it did in fact change color. All right, so hopefully you can see now, this has changed to a light green color, right? We went from 0 to about 100. So that's how these test strips work. They're testing for the presence of glucose. If glucose is present, it's going to change color from this yellow to one of these shades of green. All right, so what I'm going to do here is in my water, I'm going to test each one of my containers for the presence of glucose before we start the lab. So I'm just going to dip each one of these in here, and I'm going to leave it right on top. And we'll come back to that in a little bit. To each one of my samples, I'm going to add some of my lactase enzyme. Oh. This one was not supposed to get. I'm going to change this around. Little mistake there by Miss Roberts. This one is supposed to get the enzyme, not that other guy. And we need to add some enzyme to our last one. Okay. So let's take a quick peek at our before colors. They're all yellow. Okay. None of our before colors have changed. So if I were to write down here, move this guy out of the way, my before color, I'm just going to put a Y in all of my before categories for the fact that it was yellow, right? And that should make sense because in all of these samples before, we just had water, we just had milk, or we just had a sugar water solution. There shouldn't be any glucose before we add an enzyme if we're trying to test for whether or not this enzyme actually breaks down these disaccharides into their monosaccharide components. So I'm just going to, I've added the enzyme, right? It was a water with some enzyme, lactase only. So this is only milk. There's no enzyme in here. This is lactose and an enzyme. And the very last one is also sugar water or sucrose and an enzyme component. So I'm going to let them sit here for a couple of minutes. I'm going to pause the video, and then when I come back, we're going to test them again to see if we've got the presence of any glucose.